So I'm actually kind of honored and it's a pleasure for me as well to be the sandwich here between Oron and a piece of plastic, if you want, and Kira and bodies. So I'm looking at, I guess, semi-livings, parts of bodies, but I'm adding another component into that, which is the movement. And movement is a ubiquitous phenomenon, which defies attempts at understanding and definition. Motion is observed by attaching a frame of reference to a body I do like that, and measuring its change in position relative to another reference frame. So in a way, movement is relative. Movement means ever change, and movement for us is visceral. So how much can a body be fragmented before it is not a body anymore? How much can life be stretched before it is not life anymore? It is not death we are talking about here, but the semi-livings. The Tissue Culture and Art Project has been exploring and actualizing this question for more than a decade. We coined the term semi-living to describe the type in-between life forms that results from maintaining and growing parts of bodies in artificial conditions. So, as Oron already talked about, techno-scientific bodies. And if Oron is into eyes, I'm into ears at the moment. And, again, what is it about cell that is so evocative? Even though life seems to defy a clear definition, and exercise in progressive reductionism will lead to the sticky area of borders of life, the level from which belows life no more, or from an opposing view, death is no longer the outcome of expiry. The level, unsurprisingly, is the most common referred to as the basic unit of life, the cell, and not the DNA. And this is another quote from A.G. Wells and Huxley. And again, in the context of biopolitics, I think it's a very important to talk about. We may compare the body to a community and the cells to individuals of which this vast organized population is composed. It is very important to realize that this is not a merely allegorical comparison. It is a statement of proven fact, for we resort here to stress of italics. Single cells can be isolated from the rest of the body and kept alive. And again, at that time, this was such a revolutionary idea. We're not a whole body. We are a community of cells that can be separated, fragmented, and kept alive. And again, not just that we can, not a closed body. We're not even a human body. We were never human. We are actually already an ecology of human and non-human. Okay, and now I will, maybe to unpack a bit of the issues, maybe I'll give you a bit of explanation about regenerative medicine or tissue engineering. And you can see here, this is taken from directly from the presentation of Joseph Vacanti, who's responsible for the creation of the mouse with the ear on its back. And on the left side, I think this is from '89, from the Life magazine. You can see the vision that science had at the time of repairing the body. And if we're talking about repairing the body, of course, we're talking about enhancing the body. And this is the idea of the cyborg. We'll take plastics and put them into the body, or the Tin Man, if you want, and repair the body. Then 10 years later, Joseph Vacanti actually discussed this kind of paradigm shift. Why not use the body own regenerative abilities that were developed over millions of years to actually fix the body? So in short, if you want, you take biodegradable polymer, create the scaffold that you want, a piece of plastic if you want, you put the appropriate tissues into it, grow the organ you want, and put it back into the body. But this is not as simple as it might sound. And also it raises another point that Ron was writing about it already in 1996 in his thesis as a designer. So think about that. If we can actually repair the heart, if you look at the cyborg or the Tin Man, and then create a heart out of plastic. So take a pump and put it into the body. Then we reverse this idea and say, well, let's use the cardiac cells, the heart, in the body and grow a heart. Why not do another kind of idea? Forget about implanting back this kind of grown heart into the body. Why not grow a pump and leave it outside of the body? What kind of relation, what kind of how are we going to actually look at this kind of semi-living thing?
1997, Boston, Massachusetts.
Sorry, I didn't know there is sound, but that's appropriate. So here you go. This is the first creation of this kind of piece of plastic body. Let's connect it together. Here we go. We have that. And again, the techno-scientific body is an animal. But then going back to the idea, why do we have to, obey the tyranny of nature, if you want? Why not then change the design and have those living surfaces growing as a chair, for example? And that was the idea, and look at it also from an ecological perspective. We will not throw this chair. We will actually water it, care for it, etc. Well, this doesn't work this way, as you all know. So I'll just give you this kind of definition. What are the semi-living and partial lives? Are a new class of object being constructed of living and non-living materials, cells or tissues from a complex organism, grown over into synthetic scaffold, and kept alive through artificial support. They are both natural and cultural, hybrids, both similar and yet very different from human artifacts such as object and, of course, are selectively bred animals and plants. And I'll let you finish reading that and say, but at the same time we are creating here another class for exploitation. Okay, Ron talked about Alexis Carrel and Niederberg. If we're thinking about Rosie, Ron discussed the chimpanzees, and I'll talk about the bonobos, and this is Honor Phil. A very interesting scientist, who actually suggested she developed tissue culture and the beginning of tissue engineering, if you want, as in England in 1910 to 30, and in the lab that was called Strange Ways, which is a very nice name. And she suggested, let's look through the tissue culture. Let's take the tissue culture point of view and look at that as a reference point. And of course, as a scientist, she said, let's be careful about that. But this is a very interesting and important exercise because tissues in a dish, as Aaron said, do not, the ideas of race, gender, age, time do not apply to them. Let's think about from their perspective. And then those are the semi-living art that we did, semi-living worry dolls, semi-living steak that Aaron talked so much about, so I'll skip. You can see the extreme Nouveau cuisine. And this is the semi-living or victimless leather. So the same idea, this kind of utopia that we can grow leather-like material without killing animals. And this was shown at the MoMA. And at that time, for the first time, we used embryonic stem cells from a mouse, and the cells just grew like crazy and actually formed a small embryonic body that clogged the whole system. The curator called us and said, what do we do? The system is out of control. What do I do? And we said, well, listen, we have to actually turn the plug off, we have to kill the thing. And that's what happened. That's what she said. I felt cruel when I turned it off, said Paola Antonelli. The it in question is that, I had to turn off this life support system and because it was getting out of control. There are two things I want to mention here. One, again, the symbolic element that this victimless leather had that people put into, people are killing more cells when they, in the morning when they brush their teeth. But here it was a murder. The second thing, that this exhibition was all about, how technology and design can actually save our future. Look at all those kind of advancements. And look what happened. We have a horrible mistake that goes out of control. This is the first victim of this, unbased utopia, if you want. Okay, I'll just quickly go over ideas about life, death, science, movement, and novelty. So until then, when we grow our cells, of course, cells have a different scale of movement and time. So people see this blob of thing in the bioreactor, and they have to believe us that it's alive. And the only time that people actually feel, they say, that it's alive is during the killing rituals, when we actually killing those kind of sculpture, either eating them, or in this case, a very humble thing of touching it. And this is the killing ritual of the semi-living ear that you can see here. Now, for me, it's very important to have this kind of object, if you want, this kind of visceral feeling toward those kind of new class of object being. And the fact that we have to kill it in order to have this kind of effect is problematic. Therefore, we're looking at movement as a way of life. And you probably all know about this invention that led actually to one of the most, biggest invention in the industrial world. And this thing was a novelty. It's not really discussed a lot. That this was more kind of a party popper that later on became utilized into becoming the steam engine as we know it today. And also as a metaphor to a body. So here we have object becomes alive. And here we have another novelty, but in the name of science. And while this is running the Medusoid, I might read you some of the statements. We found the engineer. We found the inspiration from the engineering solutions that are found in nature, and we use them to develop new technologies. In this project, we actually had a slightly different approach, which was to develop new tools that will allow us to study engineering solutions that nature never came across, to find out the solutions that were missed by evolution. And this is actually rat cardiac cells growing on a PDMS, on a piece of plastic, and through electrical simulation, they are moving. The project is just the beginning of what we see as a reimagining of the synthetic biology, not just on the molecular level, in this case, but really as a recreating of an entire organism. Now, one of the people who read about that said, while the Medusoid, think about the name, Medusoid, is an interesting advancement in cell culture, it can't be called an animal any more than a cell layer growing in cell culture dish. I am startled that the staggering exaggeration of the author, quote, we took a rat apart and rebuilt it as a jellyfish, was not commented by the article's author of Nature editors. This is in Nature, yeah? Quotes like that enter the mainstream media and deliver a completely wrong picture of the state of science and scientific result to the public. But beyond that, and I think it's the movement, again, this kind of movement that creates that. What is the Medusa, as we know? She's a female, a monster, a protector, laborer, bearing of life, hideous creature. So what we're trying to say in this case, if we're looking at things become alive through a novelty such as the steam engine or this Medusoid or even the incubator, that such cases when there is liminal life, in order to actually have some kind of classification for them, we have to articulate them very aesthetic and to large extent public imagination. And we try to do that with actually very crude way of sticking our pig wings into a heart beating, and you can see the movement here. But then we decided to go a bit further because we work a lot with scientists and engineers. And this is, how much do I have? How much time? I still have time? Cool. Okay, so I'll give you a bit of biology if you don't mind. This is a skeletal cell, a skeletal muscle cell. So skeletal muscle cells are very interesting because they start with one nucleus cell, as you can see, and then in culture they become myoblasts, they're called. Now there is a certain procedure that you can do. Basically you starve them, and as a result they create those kind of cells with multi-nucleus. And those are myotubes that then becomes myofiber. And these are the skeletal muscles in our body. Now, once they become those myofiber, it's also they cannot reverse back. They are on the way to death, if you want. They matured, and that's their end. And that's what you get. And when you look down a microscope in a dish, it's quite amazing to see those cells suddenly twitching. So what we are trying to do, basically, we would like to use them as actuators. In order to actually move this kind of movement to the audience, we need to align them together and use their, if you want, movement to create this kind of, as Ron said in the talk about the state, we have to create these little genes for them to actually work and move and twitch. And we work with engineers. I don't know if there are any engineers here, but usually engineers salivate when I show this kind of slide. And this is, this is actually how we built it together. And there is an explanation. I won't even go into that. But what I can tell you about that this is a piece of over-engineering because it never worked. So we said, okay, let's go back and try and think about in a more simple terms so we can actually put cells into it. And that's one of the things that they came upon. And it's still in progress. We're still working on it. I think what I have to tell those engineers is let's think soft and pink, and maybe that will help a little bit. But I might finish with this kind of statement, and we can move on to the question and answers. To end, our proposed muscle tissue actuator is a tangible, visceral example of a contemporary liminal being, or what we can be considered bare life. Created through new scientific understanding and technological development, it is also a machine with a potential utilitarian value. Where and how it should be rendered within scientific and cultural taxonomies is the major aim of this project. Thus, from a conceptual and scholarly point of view, we seek to combine the tension surrounding bare life machine dependency, such as in the case of incubator or medusoid, as you know, and movement as a life sign, such in the steam engine, to generate discourses of new relations and empathies with different gradients of life for a more visceral understanding of the human position within its environment. This will be done through aesthetic and public engagement, which enable articulation of meanings toward imagined future. And I use Foucault because it's Biopolitics. There's no one author, but rather a group of people. Thank you.
These the lights. I think we have about 15 minutes, for
yeah, we do definitely have 15-20 minutes on this. We will take questions and answers. This work, I don't think we can ever quite prepare ourselves. The kind of controversial nature of this, the absolute cutting edge, the shocking and really stimulating components in their work. It's really remarkable. I ask you, do you have questions? Please.
Aaron, I like how you spelled out like the precisation of DNA and of it being the real thing. But then at the same time I thought about all these genetics engineering companies that are selling genetically modified crops that have to go tell long stories that the DNA won't go into other species and it won't go in other plants. And if it happens, it will replicate because they build in something that this won't happen. So there the DNA is not counting at all. It's the whole organism, it's the right chemicals to spray on it and so on and so on. Do you have any reflection on this kind of two trends within genetic engineering?
Yeah, and it's actually quite interesting. Let me maybe talk about it in some different way. Colleagues of ours, and I think we heard about him in one of the papers, Critical Art Ensemble did a work which is called The Cult of the New Eve. And what they've done there, they were looking at the rhetoric, especially in the 1990s, the birth of kind of the big push towards genetic, or the second wave, because there was the wave in the 70s that didn't really work out, and looking at the rhetoric that was used in order to describe what was going on, and they realized the rhetoric was a religious rhetoric, and they decided to create a cult which was based on the type of statements that came out of the people. And what we see now is actually the opposite through synthetic biology. We're going back to this because we forgot about the Second World War. We forgot about the idea of the creation of life as a problematic thing. So this kind of oscillation between different strategies of describing is something which is quite apparent within kind of contemporary biotechnologies anyway. In regard to specifically to what you're saying, that's not really the case, because they would talk about, and in Australia we have it at the moment, because Australia is one of the last places that is just now allowing the introduction of GM crops, or actually they allowed stuff which wasn't kind of for human consumption, like cotton, although they produce cotton oil for human consumption, but that was kind of hidden from view. It's again this aesthetic of disappearance. And this type of rhetoric appears, although to such an extent that we had a Minister of Agriculture in Western Australia from a Conservative government a few years back that said, if we eat genetically modified crops, we might grow tails. And he was talking about kind of drawbacks, and everyone was against him. So then it was, the technology is powerful, but it's not as powerful. And it's really about kind of oscillating with public anxieties. I think, again, not speaking directly to your questions, but speaking to my experience working within the field of synthetic biology at the moment. I'm involved, as we heard, in not synthetic atheistics, but synthetic aesthetic, and it is an initiative which was paid by, or was developed through a sandbox event that was organized by the National Institute of Science and Health, or whatever, and basically they put a pool of money, and they grabbed about 50 top synthetic biologists, said, okay, bid for where the money should go. And Drew Endy, who was one of the main prophets of synthetic biology, the one that developed the iGEM and the registry for standard biological parts, and social scientists did the dance based on the golem and got the money to get artists and designers to come and work in promoting synthetic biology. And I was one of the artists that was selected, and the reason I wanted to go there, I wanted to see the belly of the beast from the inside. And what's interesting there is this idea of trying to find a way to capture the public imagination in a way that they say, learning from the lessons of genetic engineering, let's not make the same mistakes. And the same mistakes, I think, is this type of thing, this rhetoric that was used, the exaggeration of the potential as well, because there's a flip side to that. I have another talk where I talk about the fact the mouse with Iron spec, for example, was used by Greenpeace and the Sierra Club to oppose genetic engineering. So this is the monster of genetic engineering. Well, there was nothing to do with genetic engineering in the creation of the mouse. So I think that both sides were guilty of exaggerating the potential and the power of those technologies. And there's kind of ways of kind of pulling back. So I don't know if I answered your question, but I'm kind of giving you some background around the whole complexity of the rhetoric that's currently being used.
I also, maybe I should give some credit to the male, gender by saying that the reason, the DNA is there. It's as important as the context and other things many times. Ron will disagree with me on that, but you have to look at the relations among the things rather than say this is not important, it's only that. So it's all the time in constant flux. So still the DNA is there. Yeah.
Yes, please.
I don't know anything about blood serum, but I was wondering, do you need to kill a calf, or can you not just bleed it or let blood the same way the African tribe bleeds their cows and mixes it with milk and then they keep living?
Because that's more humane, huh?
No. Okay.
But there is okay.
Blood serum.
No,
I agree with you, and it's possibility.
No, it's not.
What?
It's
because of the
antibodies.
The antibodies.
Okay. Just to explain, the reason why you use fetal calf serum or fetal serum, the fetal is the important thing.
It hasn't been born yet.
Exactly. So it wasn't exposed to the environment, it didn't develop antibodies, and therefore it won't reject the cells. Yeah. So you really need to get it. And what they're doing, and actually, we've done quite a lot of research. We had researchers at Symbiotica that did research into the origin of those fetal calves. The rhetoric that is being used is that those calves are coming from cows that happen to be found pregnant where they're being slaughtered. which I find with industrial farming a very strange thing to do, because cows never have sex. Yeah. So how, it's the immaculate conception of the... I just don't get it.
I've lived a farmer.
So, and there's companies that supply it, and obviously they have to supply it. They can't rely on chance that they find a pregnant cow and they slaughter it. So I would imagine that those cows are deliberately being impregnated for the production of fetuses for fetal calf serum. And there's kind of lots of, and the scary thing is that actually what they do, they open the cow out, they stick a huge syringe or a pump into the heart of the calf and suck all of the blood out while it's still alive.
Like that scene from the Tarantino film, it's like adrenaline in the heart.
Exactly, but that's the opposite, sucking out all of the blood. So, whoever's... the problem is also that obviously scientists are really, they would love to reach an alternative to fetal calf serum. The problem with that, it's non-defined and it's not standardized. So it means that every batch of fetal calf serum is different, and that affects the results of their experiments. But for the last 50 years, attempts to try and find solutions and replacement for fetal calf serum, especially for high metabolizing cells, if you want to grow large amount of muscle cells, for example, you really need to pump them up with fetal calf serum. You actually start with 20%, you drop it to 10, and then you drop it to 5 to differentiate them. There's no solution, and that's why I say when you follow the rhetoric of what's going to happen in the next 2 weeks to do it in vitro meat, you would be surprised by how we are being deceived.
And do they have a musical preference?
The cows?
No, the cells that you're growing. I mean,
we did.
We did,
with
yeah.
Adam Zaretsky,
Yes.
we did play music to cells for vibration. Yeah, in his project, it was in the MIT. I think Humperdinck. Yeah, killing
that's a monster.
softly. Yeah,
No.
the best. Yeah.
That's an Adam Zaretsky. We should be credit to that. It was a way of stressing bacteria to such an extent that they develop some kind of antibiotics, but not stressing them too much to kill them. And he tried motivational tapes, nothing happened. He tried heavy metal, he killed them, and then Killing Me Softly by Humperdinck was the one. But what we done with him was to play pig music to our pigs, to our pig wings. And we usually don't do it, but in this case we decided to play scientists, and we actually did sections of those pig wings that we played the music to, and we found that those we played music to actually had a much more, we're using bone marrow stem cells, which we differentiate to bones, and they actually looked more like bone than those that we didn't play music to. So,
yeah.
We have time for, I think, one more question. Dan?
You mentioned a lot in your talk about structures and the structure around which the cells are grown hugely influence the final product. I'm just curious, I suppose maybe you could talk a little bit about those experiments or those systems whereby an existing natural structure is used to support the growth of life. So I'm thinking more specifically about, say, a large-scale manufacture of spider silk, where they take a sheep or a milk-producing animal, they put a small piece of genetic code into that same embryo, and then reimplant the embryo into a parent sheep to bring that embryo to birth. I mean, I don't know how successful those experiments have been to date, but the point that that idea of taking an existing natural structure and using it as a system to grow something, for want of a better way of putting it, unnatural. Within that, is that something that you've encountered much, or is that
Decellularization?
No, it's I think you were kind of confusing a few different things. Yeah, there are natural materials, and I think Kira is going to talk about silk. Are you going to mention silk? Yeah, you mentioned silk. but there's what's called now ghost organs, and what they do, they take organs, they use detergents to decellularize them, so basically remove all of the living or the biological cells that are growing, only leaving the extracellular matrix, so the collagen or whatever other matrix the organ is made from, and then they seed it with new cells, and those cells get a cue from the structure to be able to differentiate into supposedly the right type of tissue that makes this organ. So it's really a spatial awareness of the cells that makes them know what to become, which is quite an interesting thing. And again, it's like the cues are totally from the outside. They're not really... There's a database, we need to think about the genome as a library, and we're not necessarily pulling every book from the library in order to read. The decision what book is being pulled out of the shelf is actually external.
Okay. Look, we're going to take a very short break, so then Senator Bocek is here, and Kara O'Reilly. But before we do, let's give an incredible thank you to Oren and Ionat Zurr.
Just can I make a disclaimer?
Yes.
We're not scientists, so don't trust a word we said.
Yeah.